Hello, my lovely anatomists and physiologists. Welcome back. We are talking specifically about the pancreas. So what we can see in this photo of our lab model is predominantly, let me just trace for you, the pancreas. So this organ is somewhat nestled underneath the stomach and here kind of in line with the top portion of the small intestine. And what we see with the pancreas is that it's going to have both endocrine and exocrine function. So when we talk about exocrine, we're talking about making secretions, putting it into a duct, special tube. And you can see that duct in the diagram. You can see it has this big, long white duct that just empties straight into the small intestine. So those secretions, those exocrine secretions are always flowing in this kind of tube directly into the small intestine, never just like out in body fluids. But the pancreas also has these special groups of cells that are endocrine cells. And that means, of course, making a hormone, releasing it into body fluids. And we talk about these special groups of cells as like pancreatic isolates, where the term isolate is sort of like tiny island, right? So it's kind of like a cute name. Now, those isolates are composed of 4 different types of cells. So we have the alpha cell, beta, delta, and then what's abbreviated as the PP cell, which is standing for pancreatic polypeptide. So let me just go ahead and write that below. If you don't want to say PP in your notes. And what we're going to see is about 20% of the isolate is the alpha cell and 75% is the beta, and then just 4% delta and just 1% pancreatic isolate. So obviously our attention is going to be focusing in on the alpha cell and then, the beta cell especially. Now, some of you already know that the pancreas is involved in glucose regulation. And so that's coming from these alpha and beta cells. So the alpha cells are going to be producing the hormone glucagon. And glucagon is going to be involved in decreasing. Sorry, I was reading my notes wrong, not thinking. Glucagon is going to be involved in increasing blood glucose levels, whereas the beta cells are going to be producing the hormone insulin, and insulin is going to be involved in reducing blood glucose levels. We're going to look in just a little bit at the homeostatic regulation of glucose. And so we'll see a little bit more specifics about what each of these hormones does in terms of how do they alter blood glucose levels. Looking at those delta cells, they're producing a hormone called somatostatin. And this hormone is going to actually have an inhibiting effect for the glucagon and the insulin. And then the pancreatic polypeptide cells are involved in appetite. And then the pancreatic polypeptide is the hormone that's being released here. And it's going to be important in that feeling full after a meal. So we'll see the polypeptide hormone, pancreatic polypeptide hormone being released after we've had a meal, and it's helping us to like feel full or satisfied. And so it's, let me rephrase that. Let me just say, let's just erase that and just say that the pancreatic polypeptide is released after a meal or when fasting. So it is going to be important in helping to suppress hunger, right? And so you can get to a point in your fasting where you can kind of like ignore that feeling of hunger. Let's go ahead and introduce this disease, diabetes mellitus, and then from there, let's talk about the regulation of blood glucose. So when we see diabetes mellitus, what we're talking about here is basically this inability to regulate your glucose levels. So diabetes mellitus is your inability to regulate your glucose levels, and this can be a genetic. This can arise because of your genetics. Your likelihood of diabetes mellitus increases when you have increased adipose tissue in your body. We can see this occurring for individuals who've had injury or individuals with immune disorders. And we can also see this if you have a hormonal imbalance. So these are different kind of contributing factors to whether or not you might have diabetes mellitus. And so there's a variety. So it could be your genetic predisposition. It could be that you have a lot of adipose tissue in your body, and then that's triggering this or contributing to this issue. It could be that you've had injury to the pancreas, for instance. It could be an immune disorder, and it could also be caused just from a hormonal imbalance. Now, there's two different types. We call it type 1 and type 2. And with the type 1, this is roughly 50% of the population who have diabetes mellitus are going to be having type 1. And in this case, they don't produce insulin. And so these are the individuals who are insulin dependent. This used to be referred to as juvenile diabetes, but it can arise in our young adulthood as well. Type 2 diabetes is where you develop insulin resistance. So your pancreatic islet cells are producing insulin just fine, but your body cells, the target cells, are not responding to glucose in the way that they should. So you're still flooding your body with all of this glucose, and the cells are just not taking it out of the bloodstream. And so you have cells that are like hungry for glucose, and also you have this high level of blood glucose. Essentially, when we are looking at having an elevated level of blood glucose, I always think about it as making my blood too syrupy. It's making the blood too thick. And because the blood is too thick, you begin to have a lot of problems and breakdown in your cardiovascular system. So you're going to get some damage to some of your smaller vessels. You're going to have difficulty getting blood flowing to your extremities, blood flowing in your eyes. And so you're going to start to see a lot of problems occurring. So we can say, let's just make a note here, when you have high glucose levels, this is the reason that regulation is so important. You're going to get damage to your retina. You're going to have an increased risk for heart attack. You're going to have potential for peripheral nerve damage. You're going to have potential for peripheral tissue damage. And then you might also see kidney damage. And again, my takeaway here is these are all linked to your cardiovascular system. So by having that thicker blood, because it's full of the sugar, it's just not moving through circulation and nourishing your tissues and organs the way that it should. Okay, so let's take a look at our homeostatic regulation of glucose. And so I have separated my page into a top and a bottom section, and I have at the top our blood glucose levels are too high, and then I have at the bottom our blood glucose levels are too low. So this is going to be a negative feedback mechanism. So remember, you should think like a figure 8. So when levels are too high, we're going to take action to bring them down. When levels are too low, we're going to take action to bring them back up. So in this case, when we have our glucose levels are high, what's basically happening in this case is when you have food entering the small intestine, this is going to trigger the release of a hormone, which we can call here gastric inhibitory. Let's see. I think I need to change how I'm writing that. So when we have food in the small intestine, we're going to get the release of a hormone called gastric inhibitory peptide, and we can abbreviate that here, GIP. And when we get the food in the small intestine, really what that's telling the body is we're getting ready to flood the body with glucose. And so what we see from there is that our pancreas is going to release insulin. And you might want to remind yourself here that it's those beta cells of the pancreas that release the insulin. This has been a final exam question in years past. And what we see is that insulin, you can use the memory device, insulin gets the glucose into the cell. So when we get that high level of insulin, what we see is that body cells remove glucose from the blood. So right away, we've decreased our blood glucose levels. Now, insulin is telling those body cells to do the following. We're going to see an increase of glycolysis so that we get a high amount of ATP production. We're going to see at the liver cell specifically, we're going to build those glucose molecules and store as glycogen. So we're going to store glucose as glycogen. Let me just rewrite. And then we're also going to increase our triglyceride synthesis. So this means a couple of things. In order to make a triglyceride, you take a glycerol and you pair 3 fatty acids. So by pairing up all those fatty acids, you don't have fatty acids for ATP production. Instead, you're going to use glucose. So that's helping to remove the glucose and use the glucose to make ATP. But also we can take that glycerol and turn that into glucose. So by pairing it up with the fatty acids, we're removing this possible source of glucose. And then we're also going to increase protein synthesis because we have all of this available glucose for all of this ATP. And so protein synthesis can be an ATP intensive process. So we're basically, when we're eating food, we should expect our body to be flooded with insulin so that the glucose can go into our cells and our cells can use that glucose essentially to make ATP. If we're in this stage where we're like eating food, then we call this the absorptive state. We're actively in this period, like absorbing nutrients from our small intestine and using those nutrients for our cellular processes. Now, the person who's type 1 is not making the insulin, so they're not going to have any way of getting that glucose into their cells without taking insulin. And the person who's type 2 is still making the insulin, but the body cells are not responding. Now, if our blood glucose levels are low, this could just be the scenario of I'm not eating anything. I've been asleep. I'm asleep, right? So in this case, our pancreas detects low glucose level. And this is going to trigger the alpha cells to make glucagon. And when we get glucagon, what we see happening here is basically we get what we can describe as like glucose sparing activities. And these things include the liver is going to break down glycogen to release glucose. We're going to see the liver convert amino acids into glucose. And we're going to see lipolysis. And when we do lipolysis, the breakdown of the triglycerides, that does two things. Number one, we're releasing the fatty acids. So all the cells now have access to fatty acids to use to make energy. And then the second thing is we can take that glycerol to the liver and then we can make glucose. So the liver has this ability to produce glucose. And so if we're breaking down our glycogen reserves and releasing glucose at the liver, if we're converting amino acids into glucose at the liver, if we're converting glycerol into glucose at the liver, and if we're telling all of our body cells to switch to breaking down fatty acids to make ATP, then we're effectively increasing our glucose levels in terms of maintaining your blood glucose levels and also providing glucose for your neurons, which prefer glucose. And this is typically occurring in what we call the post-absorptive state. So you should consider absorptive state is when you are actively eating food. Takes about 4 hours to digest a meal. So most of us, if you have a breakfast, a lunch, a dinner, a couple snacks in between, all of your waking hours, you're probably in this absorptive state. What I've totally spelled wrong. And then after you're done eating food, you're not eating food anymore. This is usually when we're at rest, really asleep. Then we switch to this post-absorptive state where we're not getting this nutrient coming in. And so in this case, we have to be very careful about our use of glucose. And that's when we're switching to these glucose sparing activities. All right, stay tuned for next video. And as always, take care of yourselves and each other.